Good morning and welcome. I'm Allison Brashear, the Dean of the UC Davis School of Medicine, and we are excited to launch the UC Davis Office of Research and School of Medicine Distinguished Speaker Series in Research and Innovation. I am a strong believer in the importance of academic medical institutions and multidisciplinary research in discovering life-saving treatments and groundbreaking healthcare innovations. The pandemic has made it even more important to translate our leading edge research to the classroom and to our patients' bedsides so that we're giving all of our patients access to the safest, most effective treatments and care. As one of the country's leading medical schools, ranking 7th in training of primary care physicians, the heart of our mission is to provide our patients with the best patient-centered care possible. Research innovations are a key ingredient to making that happen. Now, it is my pleasure to introduce UC Davis Vice Chancellor for Research, Prasant Mohapatra.
Thank you, Dean Brashear, and welcome everyone. It gives me great pleasure to introduce our guest speaker for today's Distinguished Speaker Series in Research and Innovation at UC Davis. Dr. Jennifer Doudna is the Lee Ka-shing Chancellor's Chair and a professor in the departments of chemistry and of molecular and cell biology at the University of California, Berkeley. She is also an investigator of the Howard Hughes Medical Institute. Her co-discovery of CRISPR-Cas9 genetic engineering technology with collaborator French scientist Emmanuel Charpentier has changed human and agricultural genomics research forever. This genome editing technology enables scientists to change or remove genes quickly with a precision only dreamed up just a few years ago. Labs worldwide have redirected the course of their research programs to incorporate this new tool, creating a CRISPR revolution with huge implications across biology and medicine. Just recently, Dr. Doudna, along with her colleague, received the Nobel Prize in Chemistry for the year 2020. In addition to her scientific achievements and eminence, she is also a leader in public discussion of the ethical and other implications of genome editing for human biologies and societies, and advocates for thoughtful approaches to the development of policies around the use of CRISPR-Cas9. Jennifer has received many prizes for her discoveries, including the Japan Prize, Kavli Prize, Wolf Prize in Medicine, and I'm sure I'm missing out a long list of prizes. In 2015, she was named by the Time magazine as one of the 100 most influential people in the world. Dr. Doudna, we are honored to have you here with us today. We have more than 2,300 registered attendees who are very interested in what you will share with us. The floor is all yours.
Thank you, Prasant, for that very generous introduction. And I want to give a special thanks to Dr. Ralph Green for the invitation to come and speak with all of you. It's a real delight to be here. And I can't tell you how excited I've been about the partnership between UC Berkeley and UC Davis, especially over the last few months. It's been just extraordinary. And I'll say a bit more about that today. So let me share my screen. And I'm hoping everything looks good. So what I want to talk about today is CRISPR and coronavirus. And I'm going to break the talk into 3 parts. I want to start off by talking about the fundamental science behind CRISPR genome editing, because to me, this really illustrates a classic example of why curiosity-driven research is so important. And for many of you who are watching, who are starting your educational course and your academic careers and thinking about what you want to do in the future, I hope to convey to you how exciting it is to do discovery research, because you don't know what is out there. And the reason that we do this kind of work is to answer our fundamental curiosity about the world that we live in, and in particular, our own place in that world as biological beings. So I'm going to talk about the origins of CRISPR. I want to say a little bit about CRISPR and genome editing and where that is going. And it's a very fast-moving field with lots of exciting opportunities and applications. And then in the third and longest part of the talk, I actually want to turn to where we are today and facing the pandemic and some work that's going on right now to apply CRISPR as a molecular diagnostic. And I think this really illustrates both the power of fundamental science, but also the importance of collaboration and partnership in doing science that brings fundamental discoveries to bear on real world problems and applications. So where did CRISPR come from? So for those of you that have heard of the acronym CRISPR, you may or may not know that it actually describes a bacterial immune system, a way that bacteria are able to fight viral infection. And this is a scanning electron micrograph that shows the surface of a bacterial cell that's being infected by phage. And this infection process, much like viral infection of any type of cell or organism, involves a battle between the host and the virus. And so in the case of microbes, these cells are growing very quickly. They have very little time to fight off the virus. And so over the course of evolution, bacteria have evolved a number of different mechanisms of defending themselves against viruses. And a number of these have been well characterized in molecular genetics over the past few decades. But there was a system of adaptive immunity that had escaped the attention of scientists until around the early to mid 2000s. And this was a system that is illustrated here in this cartoon, in which bacteria are able to adapt in real time and acquire immunity to viruses as they infect these cells. And very briefly, the way this works is that cells possessing a CRISPR immune system have a DNA sequence in the genome that is quite distinctive. And in fact, this was one of the first tip-offs to bioinformatics researchers that bacteria might have a system like this, because they noticed a pattern of repeat DNA sequences shown by these Rs that flanked inserted spacer sequences in the genome. And these short spacer sequences turn out to be derived from bacteriophage, the viruses that infect these types of cells. And so this was very interesting. There were 3 papers published back in 2005 that reported this observation. They were not in fancy journals. They were published by groups working in the dairy industry and in the food industry, who were trying to understand how this bacterial defense mechanism might work. And what emerged over the next few years is that cells with this type of sequence in the genome are able to use it as a mechanism for defense against viruses. And the way it works is that these cells make an RNA copy of the CRISPR array. So a transcript shown here, they process that RNA into short units that each include a sequence derived from a virus, shown by the colored bars. And those RNA molecules assemble with one or more CRISPR-associated, or Cas, proteins to form an RNA-guided surveillance complex that can search the cell looking for matching sequences, typically double-stranded DNA. And when a match is found, allow the CRISPR-Cas protein to cleave the matching DNA sequence. And this is a video that just illustrates how we imagine these systems working. So here are some bacteria growing in a biofilm, let's say, and upon infection by a virus, if the cell has a CRISPR sequence in the genome, it can integrate a sequence derived from the virus into the CRISPR array. And this integration process maintains the pattern of repeat sequences that flank these inserted viral sequences. So that marks the locus as a special place in the genome from which a CRISPR RNA transcript is generated and then processed into these unit length RNAs. And in some systems, these RNAs, these CRISPR RNAs, combine with a second type of RNA called tracer and a protein called Cas9 to form the RNA-guided surveillance mechanism for this immune system. And these proteins then search the cell looking for matching sequences of DNA. When they find a complementary sequence, there's a cleavage reaction that's triggered in the matching DNA. And in bacteria, those double-stranded DNA cuts trigger rapid destruction of the viral DNA. So it's a really nifty way that cells can adapt and become immune to bacteria in real time, and then pass that protective sequence information on to their progeny. So in the course of studying this immune system with my colleague Emmanuelle Charpentier, we realized that in eukaryotic cells, in animal and plant cells, human cells, double-stranded DNA breaks are handled very differently than in bacteria. Namely, they're recognized and repaired by mechanisms illustrated here, either through non-homologous end joining that leads to typically a small sequence disruption in the DNA at the site of the break, and also to homology-directed repair if a donor DNA molecule is present in the cell and can be integrated at the site of the double-stranded break. And this was work done by many labs over the previous 2 or 3 decades to understand these different pathways for DNA repair in eukaryotic cells. And along the way, scientists had recognized that you could harness these repair mechanisms for genome engineering if you could trigger a double-stranded DNA break at a desired place in the genome. And that's where CRISPR-Cas9 comes in. So in the research that we did together with Emmanuelle Charpentier's lab and our lab members, Martin Jinek and Chris Chylinski, they figured out that CRISPR-Cas9 is a dual RNA guided protein. It uses the CRISPR RNA molecule here for interaction with a 20 base pair sequence in double-stranded DNA. And importantly, this protein also requires the tracer RNA, which is important for stable assembly of this surveillance complex. And Martin Jinek in my lab, a wonderful biochemist and structural biologist, realized that he could link together the 2 natural RNAs in the system to form a single guide transcript that had the targeting information on one end and the structural handle for Cas9 assembly on the other end. And for us, when Martin did these experiments with the single guide RNA and showed that we had understood the mechanism of Cas9 programming with RNA and that we could control its cleavage at different sites in DNA by simply changing the sequence in the single guide RNA, this was for us that kind of proverbial moment when a curiosity-driven project morphed into a project that we realized was going in a very different direction because of the exciting potential for this to be a powerful tool for manipulating DNA sequences with a precision and a simplicity that had never before been possible. And so this was really the, kind of our transition from, studying these systems purely for the purpose of understanding fundamental biology in bacteria to recognizing that we were onto a very exciting, powerful technology for genome editing. And just to illustrate briefly, again, how we imagine this works in eukaryotic cells, that of course in eukaryotes, the DNA is in the nucleus and it's packaged as chromatin. And amazingly, the CRISPR-Cas9 enzyme with its guide RNA is able to enter the nucleus with a nuclear localization signal that's been engineered onto it and search the genome for a matching DNA sequence, matching the guide RNA. And when that match occurs, we know the DNA melts in the inside the protein. The protein is able to cleave the DNA at a precise position, making a blunt double-stranded cut. And then the ends of the DNA are handed off to repair machinery in these cells to fix the break by introducing a small change, as in this example, or by integrating a new sequence of DNA if homology-directed repair is implemented. And so this has become a powerful way to manipulate genomes of basically all types of cells, plants, animals, and of course, human cells, with lots of exciting opportunities and implications for research, but also for very practical kinds of applications of genome editing that I'll say a little bit about. And in particular, a lot of attention has focused on how one might imagine using this kind of technology to correct disease-causing mutations at the source. And one of those types of very well characterized genetic diseases that has been known for a long time is sickle cell disease, which is a great example of how somatic cell genome editing could be impactful in clinical medicine. And this is just a cartoon that illustrates the sickle cell mutation. So this is a single mutation in the human beta globin gene that gives rise to mutant form of hemoglobin that is prone to aggregation. And so in patients that are affected with this disease, they suffer from cells that have this classic sickled shape, and they go through crises in their disease in which they are subject to severe pain and certainly to tissue damage. And with the CRISPR-Cas9, we now have a technology that in principle offers the potential to cure this type of mutation and disease at its source by making a correction to the genome, either by directly altering the sequence of the affected gene, or by activating the expression of fetal hemoglobin, which is also a strategy for dealing with sickle cell disease. Very exciting. This is now no longer just an idea or a proof of principle experiment in the laboratory with cultured cells, but in fact has gone into clinical trials. And over the last year, we've seen multiple announcements, including one for a patient named Victoria Gray, whose sickle cell disease has been effectively cured using CRISPR. Very exciting sort of advance for the field. And I think many of us feel the encouragement and the potential for this to have a real impact on many people who are affected by genetic disease and even rare disease, for which this kind of technology in principle offers a personalized approach to treating or curing their disease. I think about this more and more in the context of affordable access, because as exciting as the announcement about Victoria Gray was, it was also clear that in the current implementation of CRISPR for genetic disease, it's simply too expensive for most people to benefit from it. And so I think a lot about this in the context of, as a scientist, what can we be doing to make sure that we are working to make this technology available to those that need it. And I think one of the keys to this from a scientific and technical perspective is delivering the genome editing molecules efficiently and effectively into patients. We need to be able to do cell type specific editing. We need to be able to make targeted genome edits in the cell populations and tissues where those edits can be beneficial. And there's a lot of innovation that is still needed in this field. So again, for all of you that are starting out in science and thinking about what you might want to do in the future, this is certainly one area where we need you. I'm excited to be working on this problem with colleagues at the Innovative Genomics Institute, where we have a very active program on cellular delivery with the goal of creating strategies that will avoid the need for bone marrow transplantation for patients that are affected with diseases like sickle cell, and will avoid the expense, of course, of that type of procedure, but also the health challenges to patients in going through that kind of a procedure. So that's something that I work on very actively currently. And the other aspect of genome editing for clinical use that I've been working on quite a lot over the last few years is thinking about safety, efficacy, and of course, responsible use of genome editing, because with a technology like this, we have both extraordinary opportunities, but also real risk in terms of safety and I would say ethical use of genome editing. And this has come up in particular with the possibility of using genome editing in human embryos. And so that's something that I think needs very active discussion and attention. And I've been working on this with our partners internationally and working towards creating a transparency mechanism so that folks that are working on research on human embryos with gene editing have a way to communicate about their work and make their work publicly accessible. So I want to turn now to talking about CRISPR and coronavirus. And by the way, I'll just interject one other thing, and that is that I didn't mention it here, and I didn't put slides into the talk about this, but I personally am also very excited about the uses of genome editing in agriculture. And we have a couple of active programs right now through the Innovative Genomics Institute that involve folks at UC Davis, partners, Pamela Ronald and others who are working on genome editing in rice and for other applications that I think will be very impactful globally in dealing with climate change and some of the challenges that are arising very quickly with that. So I won't say more about that here, but I do want you to know that that's a very exciting area that I think is also one that will be impacted by the CRISPR genome editing technology. Now with the coronavirus, so I want to point out that CRISPR-Cas systems in bacteria are widely diverse. And this is something that's been quite interesting just from a fundamental science perspective. A lot of the work on this that I have access to is through the research of Jill Banfield at Berkeley, a very talented scientist who has been working for a long time on metagenomic sequencing of bacteria, particularly environmental bacteria. And then also, of course, with our collaboration with Emmanuelle Charpentier on CRISPR-Cas9. And so we find that these systems are diverse. They have diverse functions. They have diverse biochemistry. All of them use RNA-guided recognition. So they're all kind of, sharing that core molecular principle of recognition. And interestingly, as we have discovered recently, both bacteria and phage can encode these CRISPR systems. So interestingly, bacteriophage, although they are the targets typically of CRISPR-Cas, they can carry around these systems in their own genomes and use them to target other phage or even potentially to control gene expression from their hosts. So I think still a lot of really interesting biology to be dissected there. And so over the last roughly decade or so in my lab at Berkeley, we have continued to investigate this fundamental biology. And as I'll show you now, every now and then, those experiments reveal things that are unexpected and were unpredicted about the behaviors of the proteins and the RNA molecules that are part of these pathways that in turn have implications for technology. And one example of this is research that was done by a former graduate student of mine, Alexandra Seletsky, when she was in the lab a few years ago. And so she was studying a CRISPR-Cas protein called Cas13 that had been reported to be an RNA targeting version of a CRISPR-Cas enzyme by this group here. And so this is how these proteins work. So they're encoded in genes adjacent to a CRISPR array in the bacterial genome. And then these proteins, these Cas13s are expressed. They bind to the transcripts made from the CRISPR locus, and they have an enzymatic activity that can produce the mature CRISPR RNAs that form these RNA-guided surveillance complexes. So that was sort of interesting. And then in work that was done by Alexandra, she figured out that these proteins have a second enzymatic activity that allows them to cut non-specific RNA molecules upon activation by binding to a target sequence. So target recognition triggers cleavage of single-stranded RNAs that are found in the immediate environment of the enzyme. And you can imagine that in bacteria, this might be quite useful during an infection where recognizing a transcript from or even a section of a viral genome will trigger rapid degradation of lots of RNAs that are in the mix and triggering either cell attenuation or even cell death if the cleavage activity is significant in these cells. And so as Alexandra was doing these experiments, we were, kind of intrigued at this kind of non-specific RNase activation. And we started talking about, I wonder, why do these proteins do this? And also, is that useful at all? And it was really in a conversation with my spouse, Jamie Cate, who is also a professor at Berkeley, who said, I think this would be a really interesting way to have these enzymes report on the detection of a sequence by designing a system where the activated ribonuclease activity, the non-specific activity, would cleave a molecule, an RNA molecule that was labeled with a quenched fluorophore pair, where upon cleavage, the fluorescent signal would be released for detection. And so we literally, did the simplest version of this experiment that you can imagine initially, which was to go to the freezer and pull out a reagent called RNase Alert that some of you might have used in the lab if you are trying to see if your buffers are contaminated with ribonuclease. And these assays are set up with single-stranded RNAs with quenched fluorophore pairs where RNase cutting releases a fluorescent signal. And you can see that sure enough, if you add ribonuclease A to RNase Alert, you get a very nice fluorescent signal. And so we did the same reaction, except using various CRISPR RNAs that were programming the Cas13 enzyme to recognize different viral targets. And these are all bacteriophage lambda sequences, and testing various both on-target and off-target sequences. And what we found is that we got very nice, triggered very nice signal detection using this RNase Alert assay down to about picomolar levels of detection. And so that was kind of cool, and sort of one of those experiments that works the first time and made us realize, okay, I think we really do understand how these proteins are working. And then Alexandra graduated, and another student came to the lab, Janice Chen, and she stumbled across another surprisingly sort of parallel type of activity in a different class of CRISPR proteins called Cas12. And these proteins are similar to Cas9 in the sense that they use an RNA-guided mechanism to recognize a double-stranded DNA target and make a double-stranded DNA cut. But in experiment, an example of which is shown here, Janice Chen found that these proteins also have the ability to cleave, in this case, a single-stranded DNA substrate non-specifically after being activated by recognition of a target sequence. And this is just using an M13 phage, which is a single-stranded DNA phage. And what you can see here is that with Cas12, upon recognition of a target like this, the enzyme is able to cleave single-stranded phage very efficiently, and the DNA is essentially shredded. And this is an activity that is completely absent from CRISPR-Cas9. We don't see that kind of cleavage of single-stranded DNA at all in these systems. And then this is just showing that this activity is indeed a target-activated single-stranded DNase activity. So here's another experiment that Janice Chen did where she allowed the CRISPR-Cas12 enzyme to recognize a double-stranded DNA target. This is a plasmid molecule And you can see that in an agarose gel, we get cleavage of the plasmid. So we get the kind of expected double-stranded cutting that we would predict for this enzyme. And then if you add a radio-labeled substrate to the reaction, you can detect very rapid cutting of non-specific single-stranded DNAs after activation of the enzyme, but no cleavage of non-specific double-stranded DNA. So this is still a double-stranded, targeted type of cleavage mechanism. It's just that the activity is able to also cut single-stranded DNA that might be found in the environment, so the nearby with the and able to access the enzyme active site. Why would cells want? Why would CRISPR systems be set up like this? Well, we speculate that this kind of activity again provides cells with an additional nuclease activity that can shred single-stranded DNA that might uniquely correspond to a viral single-stranded phage genome and not harm the host chromosome because it doesn't impact non-specific double-stranded DNA. And so again, Janice, thinking about sort of how this kind of activity might be useful as a detection mechanism, designed an experiment to actually try to distinguish between the viral strains that infect humans in the case of human papillomavirus. And so this is a sequence shown sort of enlarged for these two most common strains of human papillomavirus, in which it was possible to design CRISPR RNAs that would uniquely detect one or the other of these viral sequences. And the idea here was to implement a protocol where we could take human samples, these are human anal swabs, quick treatment with proteinase K to release in this case, double-stranded DNA. This is a DNA virus. And then a quick isothermal amplification step to amplify relevant sequences, followed by detection using the CRISPR-Cas12a protein. And again, very similar to what I showed you for Cas13, using fluorophore labeled single-stranded DNAs in this case as the actual detection reporter. And so we collaborated with Joel Palevsky at UCSF, who runs a clinic in Africa. He had collected many thousands of human anal swabs and had access to these in his laboratory. He very nicely had already done a lot of work detecting these viral strains using the polymerase chain reaction. And so we simply took this CRISPR-Cas assay and asked whether we could distinguish between these viral strains and compared the results to those derived from PCR. And we were very excited to see that this system worked extremely well, not only to detect the virus, but also to distinguish between these viral strains. And so this was really the start of what Janice Chen ended up deciding to focus on for her career, at least so far, which is to develop this technology as a robust diagnostic for viruses and other kinds of nucleic acid sequences where you would like to be able to distinguish between two different types of sequences or more than two. And so in my own lab at Berkeley, I was delighted to, both Alexandra East-Seletsky with Cas13 and Janice Chen with Cas12 had done great thesis work and finished their PhDs and had left the laboratory. And Janice Chen founded a company called Mammoth Biosciences to continue developing the diagnostic work with Cas12. And I had largely, pivoted my academic lab to other projects. But in the early part of this year, when it was clear we were facing a real public health emergency with this SARS-CoV-2 pandemic, we asked ourselves, what can we do as scientists and with our expertise to address this emergency? And certainly one of the things that we wanted to focus on was testing, because it was quite clear and still is that that's an urgent need. And so we've, over the last number of months, been working on developing a better diagnostic test using CRISPR-Cas proteins and thinking about all of the parameters that are shown here that are very hard to optimize all at once, but certainly some of which are uniquely accessible using CRISPR technology. So ideally, you would love a diagnostic test to be sensitive, to be accurate, to be fast, and to be cheap. And then, of course, in the current environment, thinking about supply chain issues has been sort of a repeated challenge as well. And so we've been working on this and in the context of a 5-lab academic consortium that was put together that is links together labs at UC Berkeley and at the Gladstone and UCSF. And so this has been a great opportunity for team science. We sort of call it the lab without walls. We had initially close to 50 people that were participating in this program. Really just everybody who had an interest in doing this kind of dropped their original projects and jumped on board. And we set this up as effectively as running like a little biotech company within an academic setting where we had subgroups that were focused on different aspects of the diagnostic to pull it all together as quickly as possible. And I want to just show you very briefly where that team effort has gotten us and where we're headed with this. And so first of all, just a quick update on Mammoth Biosciences, and I'm a co-founder of this, so I want you to know that, but I've been really excited about the work that Janice Chen has been doing and another former graduate student of mine, Lucas Harrington, who together were the primary founders of this company and have really been the leaders on the scientific side in terms of developing CRISPR-based diagnostics. And right now they have, this is a slide I was kindly given by Janice Chen. This shows some of the data from their own research at the company, and a comparison here on the right-hand side with a number of different commercially available or commercially developed tests for the SARS-CoV-2 virus. And you can see that the Mammoth kit is looking pretty good in terms of sensitivity. And we're actually excited to be able to work with Mammoth to provide a beta test center for this test, which is going to be available, I think, in about a month as a laboratory test that can be run on robots. Why are we able to do that? Well, here's where I owe a huge debt of gratitude to Ralph Green and colleagues at UC Davis, because early in this emergency, back in the early part of this year at UC Berkeley, we were able to put up a quick, pop up a clinical testing laboratory with a CLIA license through our university health services and with medical supervision by Dr. Green. And so this was very important for allowing us to get testing going in the Berkeley community, both on our campus and with our community healthcare partners. And more recently, we've actually been able to get our own independent CLIA certification from the state of California. And we've recruited a medical director, Dr. Petros Jankopoulos, who's taken over for Dr. Green and now manages our medical testing that we're doing at the lab. Because of the access now to this medical laboratory and facility, we're able to run FDA tests. We can report test results back to individuals and to physicians. And we're excited to test the CRISPR diagnostic in this context so that we can do side-by-side comparisons with a robust PCR assay to see just how good these CRISPR diagnostics really are, and of course then work to improve them as needed. In our work with the academic consortium, we've actually focused on the Cas13 protein. So Mammoth Biosciences is working on Cas12, which is the enzyme that detects DNA. So with an RNA virus like SARS-CoV-2, we thought, well, we'd actually like to see if we can get this direct detection of RNA using Cas13 working for this system. And we reasoned that advantages of this would be that it's fast. It could be quantitative because it doesn't require a separate amplification step in principle, of amplifying the target sequence or converting it into DNA before detection. And it's simple. And so we put together this academic consortium, 5 laboratories, our group plus the groups of Dan Fletcher, who's in bioengineering Patrick Shu, who's bioengineering and molecular and cell biology, Melanie Ott, who runs a lab at the Gladstone Institutes, and Dave Savage in molecular and cell biology. So this has been a really fun group of diverse experts who kind of jumped on board and put together this team very quickly, this idea of a lab without walls. And right now, we're working on developing what we call a point of care test that would be useful on site at Berkeley and at the Gladstone. And if it works out there, of course, we'd love to see it useful at other institutions as well. And this is all being done with a nonprofit focus. The idea is simply to make these tests and make them available to those that would need them at cost, and to provide what I consider to be an alternative for surveillance testing, for testing asymptomatic individuals to catch those that might be inadvertently infected and spreading virus without recognizing that they are infected. So I'm going to describe three different formats of this test that we're using right now, and we're pretty excited because these have all come together in parallel and are being worked on by different subsets of this large team. And the first format is in a way the simplest, and the idea here is direct detection with Cas13. And the strategy is to take Cas13 proteins that are programmed with different CRISPR RNAs designed to recognize a number of different sequences within the SARS-CoV-2 genome, and then to combine those into a sort of a one-pot assay where activation of these enzymes leads to reporter cleavage and release of a signal that can then be detected in a sort of a desktop, very simple type of detection device. And this is where the Fletcher lab comes in, because they've been working for a long time on how to do this kind of detection, and they have a cell phone based mechanism of doing so that we're implementing right now for these assays. And I just wanted to show you a little bit of these data. So this is work done by Parinaz Fozoni, who's a postdoc with Melanie Ott, and they have just posted a paper on medRxiv if you're interested in the details here. But bottom line is that we found quite early on that combining multiple guide RNAs was an effective way to increase both the speed and the sensitivity of this type of detection. And you can see an example of that here, where including just one or another CRISPR RNA, number 2 or number 4, gives a certain rate of fluorescence detection above background. But by combining the two in these tests, we could get a much faster release of the signal. And then by comparing, doing these LOD types of experiments and looking at numbers of copies that could be detected per microliter, in these tests, we're typically somewhere between 100 and 1,000 copies per microliter. And so this is somewhere between 100 and 1,000 fold less sensitive than what the best PCR tests are typically detecting. So potentially appropriate for detection of highly infectious individuals. And in fact, Melanie Ott has been using samples from the BioHub at UCSF to show that you can definitely use the system to detect the virus in samples from highly infectious individuals, but not a test that would be appropriate for detecting very low levels of virus. So the Ott and Fletcher team is working right now to implement this in a point of care device. And meanwhile, we've been also investigating 2 other formats for CRISPR tests. And the second one is shown here. And this has been primarily work led by Patrick Shu and the group working with him. And the idea here is to implement a quick strategy for amplifying the target sequence and then turning it back into RNA using an isothermal strategy called LAMP. And briefly, the way this works is that one takes a sample. In this case, we're using saliva, and it goes through a quick lysis procedure. This is typically just heat and protease, and then subjects the sample to this LAMP procedure in which the RNA target can be quickly converted into DNA and amplified. And importantly, the conversion includes T7 promoters that provide for the ability to convert this back into RNA afterwards using T7 RNA polymerase. Now, one of the challenges with LAMP, and you can see it sort of in this plot shown up here, is that it's prone to signal that comes up independent of the template. And it's fairly well separated in this example, but in some cases, the signal can come up quite quickly, even in the absence of a template. And so for commercial tests that are using LAMP, this is one of the challenges that those tests have, is that they can lead to false positives. And so this is where the Cas13 detection enzyme comes in and provides a really nice advantage, because by turning this amplified DNA back into RNA with T7 transcription, and then using the Cas13 enzyme for specificity, and using it to detect only those RNAs that actually contain the targeted viral sequence, it's been possible to improve quite dramatically on the specificity of LAMP with the addition of Cas13. And so this is what Patrick and his group have been doing, and the limit of detection they're getting is a little bit better in terms of sensitivity compared to straight up direct detection with Cas13. And so this is a strategy that, again, we're working on currently to implement in the point of care device developed by the Fletcher Lab. And then the third format is one that I think, really I want to give a huge shout out to various members of my own laboratory who were thinking hard about the fundamental biology of these Cas13 systems. And they realized that there might be a way to amplify the activity and the signal of Cas13 by taking advantage of a natural mechanism in bacteria that does that. And I'll show you this very briefly. So this is a cartoon that illustrates what I showed you before for Cas13. So we have transcripts that are being targeted by the CRISPR RNA and Cas13, and that turns on this trans cutting activity, and you can start cleaving fluorophore labeled single-stranded RNAs, and that releases a fluorescent signal. But it turns out that one can amplify this when in a fashion that we call the nuclease chain reaction by hooking it up to another enzyme called CSM6 that is a ribonuclease naturally found in some of these Cas13 containing systems in bacteria. And briefly, the way this works is that in bacteria, there is enzymatic production of these very short oligonucleotides that look kind of like this, that will bind to an activation site within the CSM6 ribonuclease and turn it on. And so that then allows the CSM6 ribonuclease to also cut RNA just as Cas13 is doing. So, and so the idea here, and this was really something developed by Tina Liu in my lab and Shruti Jachinwal, was to add what they call a secondary activator in our diagnostic reaction that's designed to be cleaved by Cas13 when it gets turned on. And that cleavage event produces the activator for CSM6. And then once this enzyme gets turned on, it starts cutting RNA. This enzyme is cutting RNA. And this enzyme, of course, can cut more of this. And so then you end up making more of the secondary activator that turns on more of these. And you can see how the whole thing kind of snowballs. And so this so far has looked really quite promising in terms of giving us a very fast detection and a relatively sensitive limit of detection. I'll show you just a couple of data points here. So this is using the Cas13 and CSM6 for direct detection of SARS-CoV-2 RNA. And in this experiment, if basically you don't have to read every line here, but I just want to point out that that we're seeing that we can detect down to about 20 attomolar in terms of copies of RNA above background that we get in these reactions. And this scales very nicely. So it's sort of you can see the kinetics of the activation corresponds to the level of initial target molecule that is present in these reactions. Importantly, this also highlights a challenge that we have that you can see here very obviously, which is that there's significant background in these reactions. So these enzymes are kind of on a hair trigger. And so if they do get going, then, very quickly, they do start to cleave the reporter and you can see release of the signal. So this is something that we continue to work on, is how to limit this background. But even in the format that it's in right now, we're pretty excited that we're going to be able to detect fairly low levels of RNA quite quickly. And you might say, well, maybe it's not that quick if you look at the time scale. Is it taking an hour to get to that answer? And that's where the Dan Fletcher device comes in. So we found that by running our samples with that device, and if you look over here on the right-hand side, I've sort of expanded this part of the graph. What you can see is that the device is so sensitive at detecting the kinetics of fluorescence release that we can very quickly start to distinguish between a background signal and what we get for this 20 attomolar sample. So we're continuing to work on this and improve it, but we're really encouraged that this chemistry seems to be working out reasonably well. Very, very simple to use. It's very inexpensive. These proteins are trivial to make, and you can make them in very high quantities. And we're working currently with a company, Waynamics, that focuses on point of care testing to develop appropriate microfluidic chips to run these different chemistries and then use the Dan Fletcher device for detection. So the pieces are coming together, we hope, and our goal certainly is we hope by the end of this year or by very early in 2021 that we will have some of these prototype devices operational and running under an IRB type mechanism so that we can do studies on site at Berkeley and at Gladstone with actual donated samples. We're using both saliva as well as nasal swabs for the inputs here, and we'll see how the data compare to the PCR assay that we run in the clinical laboratory. And that's it. So I really want to just give a huge shout out to an incredible team of people. You can see this picture is taken quite recently. This was actually taken on October 7th, which was an exciting day for our lab, and the first time that many of us had been actually together in quite a while, all together. And we were trying to be appropriately masked and distanced, et cetera. But I just want to really thank Gavin Knott, Brittany Thornton, Dylan Thomas, Jenny, Connor, Abby, and Enrique. All of these folks just rolled up their sleeves and jumped in and started working either in the clinical laboratory or to develop the CRISPR diagnostic, even though I don't think in any case was that their original project. They just recognized what needed to be done, and they just started doing it, which I'm very grateful for. Shruti and Tina and Noam are three very talented postdocs who have been developing this nuclease chain reaction under the direction of Dave Savage and others at Berkeley, and working with folks in our lab. I really want to acknowledge my entire lab, just a wonderful group of people to work with, and we're excited to, I normally have about 10 to 12 undergraduates doing research in my lab at any one time. Sadly, we're not able to do that right now with the density restrictions at Berkeley, but we do hope to be able to welcome our undergrads back to the lab in the coming months. And then Jill Banfield at Berkeley, incredible scientist, very wonderful colleague to work with on all aspects of fundamental CRISPR biology, as well as shout out to Emmanuel, with whom we started the CRISPR-Cas9 work now many years ago. And then I'll just also point out thanks to our funding sources, as well as to Adgene, that continues to distribute reagents for CRISPR at cost to nonprofits around the world. And they've played a huge role, I would say, in really rolling out this technology for everybody's access. And I'll stop there and thank you, and happy to take any questions.
Thank you so much, Dr. Doudna, for that inspiring presentation. I'd like now to introduce Dr. Angela Haksu, the UC Davis School of Medicine Associate Dean for Translational Research and Director of the Lung Biology Center, and the Ralph Green, Professor of Pathology and Laboratory Medicine, who will moderate our discussion and answer session.
Thank you, Dean Brashear. It's been a great pleasure joining you, Vice Chancellor Mohapatra, Dr. Green, and our distinguished guest, Dr. Doudna. It's been a terrific presentation, and I have been reading the questions while you were talking, so I would be able to combine them and instead of reading them up one by one. I think it is Dr. Green's turn to start with the questions.
So thank you, Angela. I'm absolutely thrilled and delighted to be here today. Jennifer, I mean, this has just been fantastic. And little did I know back in March of this year when I began my collaboration with you and your group, amazing group of scientists, that we would have the opportunity to host you here today in what has been an absolutely spectacular hour. Thank you so much. We'll begin our question and answer session with questions from each of my colleagues and some questions that have come in from the audience as well. So our first question is from Vice Chancellor Mohaptra. Prasant, why don't you kick off?
Yeah, thanks, Ralph. Very exciting talk, Jennifer. So let me start with a very general question. Given the public's general perception about genetic engineering, do you think that medical or agricultural applications of genetic engineering will be more readily accepted?
Good question, Prasant. I have to say, strangely enough, and I don't know if this would surprise you or not, but I actually think medical uses will be more easily accepted than agricultural. And it's been a very interesting thing. I didn't know very much about these sort of ethical discussions before diving into this over the last few years, but it's clear that there's kind of an emotional, I think, reaction to thinking about food being manipulated in ways that people maybe don't understand or don't trust or feel are being done for commercial reasons. That I think has really been challenging for the whole sort of agricultural applications of genome editing, and that includes for CRISPR. That in an interesting way are viewed differently when turned to biomedical uses, where I think people in many cases feel desperate for treatments for their kids, especially feel maybe, at least in some cases, more trusting of the process of testing and review and safety considerations for clinical uses. So that's how I kind of have seen things evolving. It's very, very interesting. That surprised me, honestly.
Yeah, thank you.
Thank you, Dr. Doudna. The next question is from Dean Brashear.
You have been widely recognized as a pioneer in scientific discovery and as a role model for women and aspiring young people to achieve great success. What advice would you give young women entering the STEM fields?
Well, I guess my advice is summarized as go for it. I grew up in a small town in Hawaii, and nobody in my family was a scientist. And so, they were teachers, lawyers, and preachers. So I was lucky that I had, my parents were certainly supportive of my interests, but I was very lucky that I had terrific high school teachers in chemistry and biology in my local public high school, Hilo High School. And then later in college and in graduate school, I had, along the way, people who were really supportive and encouraging at moments when I needed it. And, by the way, for all the students listening, things haven't always been smooth sailing, as they never are, right? There's lots of experiments that don't work out, or, all kinds of things that don't go as you hoped or predicted. And that's when you really need good mentors. So that's my other piece of advice, is to really look for supportive mentors, and they can be anybody, men, women, it doesn't matter, right? It's really just people who are going to see your passion and support you at times when you need that.
Thank you very much. So there were, I know that you gave a wonderful presentation about the diagnostic capabilities of the CRISPR-Cas technology, but there were a lot of questions in regards to whether there would be applicabilities for treatment, especially for treatment in infectious diseases like COVID-19, for example. And would you care to comment on this, just to speculate?
Right. Well, these are great questions because a priori you might think, well, CRISPR is an immune system after all, so why can't we use it that way right now in humans? And I think that in principle one could. I think the challenge comes back to something I mentioned in the talk, which is delivery. So, you know, being able to introduce the CRISPR molecules into cells where it's needed is, you know, it's a challenge for therapies, including for acute therapies like treating a virus. And so in the case of coronavirus, how you would you'd be able to get these molecules into, you know, lung epithelia, for example, is, you know, it's a non-trivial problem. So I personally feel that at least for this pandemic, our best bet with CRISPR is probably using it for detection. And but who knows, you know, going forward, I think, you know, certainly as delivery strategies continue to advance and develop, it may become possible in the future to have, you know, some mechanism of effective and efficient delivery to cells that would make it a useful therapy. I think it's unlikely to happen for this pandemic.
So another question, Angela, if I may, I'll just want to put another question to you, Jennifer, and that is, you've shown us very nicely that the techniques of CRISPR and all of its related methods that you've developed to detect COVID-19 viral RNA has enabled the development of very rapid specific detection methods of the virus in perhaps less than an hour. As a person who's involved in diagnostic medicine, I ask you, if this is applicable to any pathogen, will this approach potentially revolutionize diagnostic methods in general?
I think it could. I mean, you know, I would never want to. I wouldn't want to use hyperbole here, but I think it could, because what's interesting about it is that it takes advantage of what CRISPR naturally is good at doing, which is detection. That's the way it functions, actually, you know, in bacteria, after all. And furthermore, it's also a system that is naturally multiplexed, meaning that it naturally has multiple guides that are, you know, searching the cell for different sequences at any one time. And so using it that way in a diagnostic setting, of course, also provides for various advantages, and you know, including sensitivity and precision, as well as detecting potentially multiple different viruses in a single sample. So I'm personally very excited about this. I think it does have a lot of potential. And I think we'll know over the coming months how well it performs in the real world, and that's really where the rubber hits the road. I think it's one thing to have it working well in the laboratory, which many groups have now shown can be done, and so that's great. But now we really need to see, how does it perform in actual clinical settings where we have patient samples. You know better than anyone, you know, how variable those can be, and just some of the challenges with that. So I'm excited about it, but I'm cautious.
Thank you.
Back to the treatment possibilities, there were questions regarding sickle cell anemia, which is monogenic disease, and people were interested in knowing whether in polygenic diseases there might be possibility. And somewhat related to this are the observed side effects of utilization of this technique on off-site um, irrelevant gene targeting. Would you care to comment about this, and what are the prospects of developing this technology for treatment, and what are the best target diseases that maybe we can hope for being affected?
Right. Well, these are really important questions. And so the first point about multigenic disease, I think there it'll clearly be harder to implement CRISPR, but I think it's coming. I really do. I think we continue to see advances in the efficiency of the technology. Already, certainly in laboratory settings, one can do multiplexed genome editing with relatively high efficiency. So I think that's very interesting. We're just completing a study right now with the lab of Alex Marson at UCSF, where we've been doing what I would call kind of single step CAR T generation, where you can knock out a couple of genes and then introduce a gene sequence somewhere else in the genome in one shot with appropriately designed CRISPR reagents. So that's very interesting for future use for cell therapies. And so that's the kind of thing that I think will be much, more widely implemented going forward as it becomes possible to to deliver these molecules efficiently into different cell types. And then that, of course, brings along with it the question about off target. And that's your second question. And I think there, the field, over the last 8 years has, really dove into this question in some depth. And certainly we know that if guide RNAs are designed that have close matches to off-target positions in the genome, that certainly, one can observe what I would call off-target editing. But on the flip side, if guide RNAs are designed very carefully and to avoid those kinds of close matches at undesired positions, and if the genome editing molecules are used in limiting amounts, then actually off targets can be quite rare. So I don't personally see it as a current bottleneck in the field, but I just think it's something very important to pay attention to, especially for clinical use.
Yes. Do you have, just very quickly, any comments on the, Dr. Kent Lloyd has been asking if you saw the news about the AgliLab results at Columbia University today, and if you could comment briefly on this.
Yeah, right. I mean, yes, I did take a look at that work. And, my view of it is, look, I mean, I think consistent with what we've seen from other laboratories, you can use CRISPR in ways where you can observe these kinds of undesired outcomes of editing, and certainly in embryos, this is a setting where I think there's still a lot to be understood just about fundamentals of DNA repair, quite frankly, from what I've seen. So, does this make me worried? Does this make me question the whole technology? No, it just, I think it underscores what we're already aware of, which is that, using it in embryos is challenging for sure, and is going to require a lot more research to really understand how to use it safely, if that were to be done in the future.
Jennifer, a question I was going to ask you that you've, in essence, partially answered already, which is, the CRISPR-Cas mechanism is quite incredible, really, in terms of its precision. And my question was going to be, is this just the tip of an iceberg? Are there other gene editing mechanisms present in prokaryotic cells that we can use? You've indicated, the Cas12a and Cas13, it's all part of a sort of general toolbox. Do you see this thing developing in time into an industry almost of various gene editing mechanisms that we can use from bacteria?
Oh, absolutely. And I think we're already seeing that. I mean, there's a whole sort of panel of companies that have been set up to mine bacterial genomes for these sorts of things, these sorts of systems and enzymes. And then of course, many academic labs that are doing that as well. And not only mining what's found in nature, but continuing to engineer things based on what nature has done to, make those proteins do things they maybe don't naturally do. So yeah, I agree with you 100%. I think we're going to see, I sort of imagine this, this whole kind of, virtual toolbox that's going to be available. And in the future, when some kind of genetic manipulation is desired, one will be able to go to that virtual toolbox and pull off the shelf the appropriate tool to do the job.
Great, thank you. Angela, I'm going to hand back over to you because I think we're reaching the end. We could carry on forever here, right?
Yes, but we have 2 more minutes. So if possible, René Solis is asking how is it known how the CRISPR DNA gets into the bacterial genome, genomic CRISPR array so quickly before the phage lysis occurs? Yeah,
that is a really important fundamental question. So a lot is known about the CRISPR integrase itself and that whole reaction mechanism, but what's still not really known very well is how it works in the context of the life cycle of the virus, right? Because as this questioner is pointing out, viral infection, especially in bacteria, is very fast, right? Especially for a lytic virus, maybe 20 minutes, right? It's quick. So would there really be time to integrate the sequence, make the RNA, make the surveillance complex, and then go? Probably not. So I think the best data that I've seen in the field right now for this is that, in bacteria, a lot of these bacteriophage are, maybe not a lot, but there's a significant percentage of those phage that are defective phage. And so what's thought to happen is that in a bacterial population, some lucky cells receive these defective phage, and so they are able to acquire functional spacer sequences in the CRISPR array and then provide protection to those cells. And so in a bacterial population, if a lot of the cells are killed off, but you have some survivors and they can pass on their immunity, then that's fine. So that's what we think happens in a natural setting with CRISPR.
Thank you so much. I'm going to turn it back to Dr. Mohapatra now for closing remarks.
Thank you so much.
You're still muted.
Yeah. Thank you so much, Jennifer, for being with us today and sharing your valuable time as well as expertise. And once again, congratulations for all your remarkable achievements and contributions to the human health and the society in general. I would also like to take this opportunity to thank Dean Brashear, Dr. Green, Dr. Hakshu, and to all our audience who are joining us today. Enjoy the rest of the day. Thanks.